Okay, I'm unmuted now? Yeah. Hello, everyone. May I have your attention, please? Thank you. So I am humbled and beyond excited to introduce our highly anticipated keynote speaker for this symposium, Dr. Michael Levin. Dr. Levin received a PhD at Harvard University and did his postdoc training at Harvard Medical School. He holds the Vannevar Bush Chair and directs the Allen Discovery Center at Tufts, working to crack the morphogenetic code for applications in regenerative medicine and basal cognition. Recent work of his includes the creation of bioinformatics and machine learning tools for discovery of models and interventions in cancer, birth defects, and organ regeneration. Now, without further ado, please welcome Dr. Levin for his talk, Biophysical and Computational Approaches to Study Decision-Making and Basal Cognition in Cells, Tissues, and Synthetic Living Machines.
Thank you very much. Thank you for the kind introduction and the opportunity to share some ideas with you. One of the things that I would like to emphasize today is that there's a really interesting relationship between AI and biomedicine, because in order to have truly transformative regenerative medicine, we are going to have to understand the natural intelligence of the body. So there's this sort of interplay between natural and artificial intelligence. And if anyone is interested in following up, you can find all of the primary papers, and you can find me at these two websites. So one of the things that is commonly known is that living systems are multi-scale. In other words, we have groups of animals function in collectives and swarms and societies. They are made of individual organisms, which are made of organs and tissues and individual subcellular components and genetic networks and protein pathways underneath that. But what's less commonly realized is that this is not only structural, but actually functional. That is, every level of a living system solves problems and exerts a kind of intelligence in some particular problem space. And in fact, all intelligences are collective intelligences. We, of course, are bags of cells, really. And it's very important to understand how this kind of multi-scale competency architecture is can be managed in regenerative medicine, in synthetic bioengineering, and so on, because there really are computational layers all the way through. So the way I see it, and what we do in my center, is to bounce back and forth between machine learning and biology. First of all, to use machine learning to try to understand and control the biological endpoints, for example, for medicine, and conversely, to use what we learn from the biology to create novel kinds of AI architectures that are based on much more fundamental and not necessarily neural principles. So the main points that I'm going to try to give you today are these. That first, I'm going to spend some time talking about the biology, actually, to try to paint in a different light what it is that we need to understand. And these are very significant knowledge gaps about something that's important called anatomical homeostasis. And try to convince you that actually biology and biomedicine is at a much earlier stage than one would think, based simply on the progress in genetics and stem cell biology. I'm going to claim that fundamental advances in new medicine is only going to appear if we understand the decision-making by cells and tissues. Not only mechanisms, but actually the algorithms of decision-making. And I'm going to show you that a key medium for that kind of computation is non-neural bioelectricity. It's basically a new kind of epigenetics, but it's a very important software layer of physiology that sits between the genome and the body, and we need to understand this. And we are working to crack the bioelectric code, which will enable a huge class of electroceuticals for all kinds of new applications in birth defects, regenerative medicine, cancer, and synthetic bioengineering. So basically, the whole talk boiled down to two sentences is this. I'm going to show you that, like the brain, your body tissues form electrical networks. And these networks make decisions about dynamic anatomy. And new tools in AI and machine learning are helping us to target the system for some really amazing control that enables us to reap some very serious benefits that have advantages over competing technologies. So I show you this five-legged frog at the beginning, just to say that I'm going to show you all kinds of weird creatures today. These are not Photoshopped. These are real living organisms that are alive and well in our lab, that serve as tests of the various theories that we develop. Okay, so let's talk about the fundamental knowledge gap. What is it really that we want to do in biology and medicine? Consider what the end game might be of all of our activities. So at some point, someday in the future, you should be able to sit down at a computer and draw, right, the anatomical layout of the animal or plant that you want. So to really specify the structure, the large scale structure of what you want to have, and not at the genetic or biochemical level, but actually simply draw the thing you care about, which is the shape of an organ or an appendage or whatever it is that we need for medicine, whether for transplantation or directly in the patient. You should just be able to draw it. And if we knew what we were doing, we would have a piece of software, which I call an anatomical compiler, which would take that description and convert it into a set of stimuli that would have to be given to cells to build exactly that shape. So here I've drawn a three-headed planarian, a three-headed flatworm, and here is the three-headed flatworm that would arise if this thing was able to tell me what to do to all of the cells, what stimuli to give all the cells to get them to build this. Now, why is this important? Well, if we had something like this, basically all of the problems of medicine, except for infectious disease, would disappear. So if we knew how to tell groups of cells what it is that they should be building, we would be able to resolve birth defects, traumatic injury, cancer, aging, degenerative disease. All of these fundamental problems hang on one basic thing, which is that we really need to be able to convince groups of cells to build whatever we want them to build, healthy new organs. Now, so let's ask this question: how do collectives of cells know what to build? So this is a group of embryonic blastomeres shortly after fertilization, and this is the cross section through a human torso. Now look at this incredibly complex order, right? Everything is in exactly the right position, orientation, the right shape, the right size, next to the right neighbors. It's an incredible amount of order, and so we ask, how does this group of cells build exactly this every single time? And so where is this information? Of course, immediately we're tempted to say DNA, it's in the genome, but of course we can read genomes now, and we know now that it is in fact not in the genome. There's nothing in the genome that talks directly about any of this. The genome refers to proteins, so it specifies the micro level hardware that every cell gets to have, but doesn't say anything directly about the shape, the size, the symmetry of the body. So we have this important question. If we wanted to repair a piece that was missing or damaged, we wanted the cells to rebuild it, what would we have to do to these cells to get them to do it again? And as engineers, we would like to know, how far can we push this? In other words, could we get the same cells to build something completely different? So let's think about what individual cells can do. This is a single cell. This is a creature called a lacrimaria. You will notice that it has no brain, it has no nervous system, it has no stem cells, no cell-to-cell communication. It is incredibly competent in its own local single cell level goals. So it handles all of its metabolic needs, all of its transcriptional needs, its morphogenetic needs, behavioral. So here you can see it hunting for things in its environment, the food. How can it do this without a brain? So that's the topic of a field, an emerging field called basal cognition. But what's interesting for us now is that these kinds of creatures, these single cells that have local goals that they're very good at pursuing, can actually work together to pursue much bigger goals. And I'm going to show you that momentarily, but here's the basic framework. We all start life as this single cell, this fertilized egg, and this is going to self-assemble into one of these remarkably complex morphologies. We know right away that stem cell biology is not sufficient. This is a teratoma. This is a tumor. It may have skin and hair and teeth and bone. And so the stem cell part has gone fine. The stem cells have made their various mature tissue derivatives. But what's missing, of course, is three-dimensional organization. So not only do you need to have the building blocks of the body, but they actually need to know where to go relative to each other. So in fundamental developmental biology, the paradigm looks something like this. It's very much based on emergence and a kind of feed-forward open loop system where you have gene regulatory networks. So genes turn each other on and off. Some of these genes make proteins that do things, they diffuse or they're sticky or they exert force. And then all of this happens in parallel. And then there's this magical process of emergence where lots of simple things happening repeatedly in parallel give rise to a complex outcome, such as this beautiful salamander. Of course, all of that does happen. So this is true, but it's very limiting because it has a fundamental difficulty, which is if you wanted to make changes here, let's say that I wanted this salamander to have threefold symmetry instead of bilateral symmetry, or I wanted more legs, or I wanted two tails or something. This makes it look like all of the changes have to happen down here. You have to make the changes at the genetic level that will then propagate. And of course, reversing this is a intractable inverse problem. We have no idea in general what changes to make down here to give us the desired changes at the system level, right? So reversing this kind of recursive, massively parallel system is almost impossible. Now, there are other difficulties, which are that we have really some basic areas of ignorance about genomes and anatomy. So I'm going to introduce you today to another creature called a planarian. And planaria, and so the thing about planaria is, as I'll mention in a minute, planaria regenerate. So if you cut off their heads and tails, planaria regenerate their heads and tails, and they have a set of stem cells called neoblasts that produce all of the new material. So here's a species of planaria with a flat head. Here's a species with a round head, and I can do a thought experiment. I can say, okay, I'm going to take half of these stem cells from this flat-headed species and introduce them into this round-headed species. So there they are, they're mixed around. Now I'm going to cut the head off, and I want to know what head shape is going to form, right? The green ones know how to make a flat head, the red ones know how to make a round head. What shape is the chimera going to make? And the interesting thing is, I mean, so here are the options, right? One of the shapes might be dominant to the other. It might be an in-between shape, or maybe no, it will never stop regenerating because neither set of cells is ever happy about the current state of the shape of the head. So the interesting thing is that despite all the papers in Nature and Science about the molecular biology of stem cells in this model system, we have not a single model that will give a prediction on this. Okay, because while we understand pretty well the individual molecular events that guide individual cell decisions, we have very poor understanding of what guides large-scale anatomical level decisions. And so how this collective is going to decide what head shape it's going to make, we actually have no models that make predictions about that at all. So overall, where things stand are that we're very good at manipulating the hardware, so cells and individual pathways, proteins, genes, but we're really a long way from form and function, and so control of form and function. And so what I've argued before is that basically, if we kind of think about the journey that computer science took, that went from this is what programming looked like in the 40s and 50s, where you literally had to rewire the system physically in order to get it to do something different. And now, of course, why don't we do this, right? When you want to switch from Photoshop to Microsoft Word on your laptop, why do you not get out your soldering iron and start soldering away? Because we've realized that if the hardware is good enough, then you can control it by experiences, by stimuli, right? By reprogramming, not by editing the hardware. And yet all the biology and biomedicine is down here right now. All of the most exciting advances are all about, CRISPR, genome editing, changing promoters and protein engineering. It's at the micro level hardware. So this whole journey is open to us in biology. We are just at the beginning of this. And I'm going to show you some of the ways in which the biological hardware is absolutely good enough. It's in fact amazing. One of the interesting things about morphogenesis is that it is reliable, but it's actually not hardwired. It's quite flexible. So here is this axolotl. It's a Mexican salamander. These guys, throughout their lifespan, regenerate their limbs, their eyes, their jaws, their spinal cords, portions of the brain and heart. So amazing. Everybody wants to be like an axolotl, of course. All of regenerative medicine is thinking about how to crank this ability up in humans. But the most amazing thing about it is that if you, for example, amputate a limb here, in fact, you can amputate it anywhere you like, it will grow very rapidly. It will make exactly what's missing, and then it stops. That's the most amazing part. How does it know when to stop? Well, it stops when a correct salamander arm has been produced. And if it's cut here, it will grow this much. If it's cut here, it will only grow this much. So it's context sensitive. It can deal with novelty, but it's reliable. It can get to the same goal from different starting positions. That, by the way, is William James's definition of intelligence, is a system that can get to the same goal through different means. It challenges to ask how this collective of cells can tell what a proper salamander arm looks like. Here are the planaria I told you about. So here are these flatworms. They have all kinds of internal organs. They have a true brain, lots of the same neurotransmitters that you and I have. And if you cut them into pieces, and the record is something like 275 pieces, each piece becomes a tiny little worm. It grows exactly what's missing, no more, no less, and becomes a tiny little worm. In fact, they're so good at regenerating, they are immortal. So don't let anybody tell you that aging is some sort of consequence of entropy or thermodynamics, that it's, we have to get old and die. That's actually not true. These planaria don't have a lifespan limit. They live forever. And there's no such thing as an old planarian. They simply keep regenerating whatever cells senesce and die. Now, regeneration is not just for so-called lower animals. So the human liver, can regenerate, and the deer, which are a large adult mammal, can regrow their antlers every year. They grow these antlers up to 1.5 centimeters of new bone per day that they grow. Okay, this is bone, vasculature, innervation, skin. And then human children below a certain age can also regenerate their fingertips. So we also have some of this regenerative ability. Of course, our bodies built all of these organs during embryonic development. The question is, how do we kickstart them? And how do we understand how the cells make decisions? Are they going to scar? Are they going to build an organ? Which organ are they going to build? And I want to kind of cement this idea of intelligence and goal directedness in the following, because it's going to be very important momentarily with this example. This is something we discovered a few years ago. This is a tadpole of the frog. So here are the two eyes, nostrils, brain, and gut. These tadpoles, in order to become a frog, they have to rearrange their face. So the jaws have to move forward, the eyes have to move, everything kind of has to remodel. And it was thought that, every tadpole looks the same and every frog looks the same, so that all you would have to do is somehow encode a distance and a direction for every piece of the head to move. And then if everybody does that, then you'll get from a normal frog from a normal tadpole to a normal frog. Well, we decided to test that, and we made these so-called Picasso tadpoles. Here's an example. Everything's in the wrong place. The eyes on top of the head, the jaws are off to the side. Everything is just completely scrambled. And what we found is that these animals become largely perfectly normal frogs, because all of these pieces, even though they start off in the wrong place relative to the wrong thing, they all will move in novel paths. Okay, not the paths that they normally do in embryonic development. They move through novel paths. Sometimes actually they go too far and they have to track back a little bit, but they keep doing it until they end up in this correct configuration through this interesting algorithm that we were able to infer. So the amazing thing here is that genetics doesn't specify a hardwired set of movements. What it specifies is a machine that can basically perform an error minimization task. You can think of it as some sort of means ends analysis, where basically it just progressively reduces the error compared to the correct pattern, which again brings us to this notion of what a correct pattern is. So in my group, what we focus on is on top of this feed forward sort of emergent picture, we put these feedback loops that talk about what happens when the system is deviated from the correct state. So now this could be injury, it could be mutation, it could be teratogens, many different things. There are these feedback loops that kick in both at the level of physics and at the level of genetics to get you back as best as it can, get you back to where it's supposed to be. This is basically homeostasis. Now, on the one hand, this should not be a surprise to anybody in biology. We know there are feedback loops. Of course, we know homeostasis. But there's a couple of things that are different here. One is that whereas normal homeostasis usually is all about some sort of scalar, pH, hunger level, something like that, and so it's a single number. Here, what you have is you have the set point for this homeostatic process is actually a fairly complex descriptor. It's some sort of coarse-grained description of what a correct anatomical pattern is. So you have to think about how could biology store something like that. That's the first thing. And the second thing is that it suggests an important change of strategy. Instead of having to make changes down here, which are very difficult to know what they should be for changes out here, instead of solving this inverse problem, what we might do is do the same thing you do with your thermostat. When you change your thermostat to a different setting, you don't rewire the thermostat. You don't change the structure of the thermostat. In fact, you may not even know how the rest of it works. You don't need to know. The only thing you need to know is the fact that it is a homeostatic system, and you need to know how to read and write the set point information. So maybe what we can do biomedically is leave the system intact. It's very good at following different set point information. Let's leave that alone. Let's exploit that. And instead of trying to alter the machine by genome editing or changing pathways, let's try to rewrite the set point. So here's what we've been trying to do. We've been trying to identify the mechanisms by which these set points are stored and learn to read and write them. And I'm going to show you how that works. Now, in this case, the set point is quite large. And one thing I want to point out is that as you know, during evolution, of course, you have these individual cells that are operating on very small local single cell set points. Then they start to work together, and there's this collective intelligence that can work towards very large goals. You know, this massive goal that's bigger than any individual cell. In fact, this is truly a collective intelligence, because during this process, many of the cells actually have to die. The cells in between the fingers have to die, and so all the cells are kind of this massive collective that's working on this giant thing, not on their own individual goals. But that process can break down, and we'll talk about this momentarily. This is cancer. This is, in fact, glioblastoma cells crawling around. And what happens is that individual cells can defect from this process. They can disconnect from this network that binds them towards these large-scale goals, and they can start rolling back to their ancient amoeba level goals, and they treat the rest of the body as just external environment. So the size of the goals that cells work on can grow and shrink during the lifetime of the animal. And there's some very interesting aspects to this. So let's think about this now. What I want to talk about is exactly how does this happen? How do cellular goals scale up, and what can we do about it? And for that, we have to understand bioelectricity. And so I'm going to do is talk to you about some methods for addressing what I think is basically the software of life. And then let's talk about what can you do with this. I'm going to show you some examples. Now, we're going to spend the next half an hour talking about bioelectricity. And I want to be clear, it's not because I think that bioelectricity does everything in the body. It's clearly one layer of all kinds of interesting signaling modalities. Of course, there's chemical gradients, biophysical pressures and tensions, extracellular matrix, all kinds of things. But bioelectricity actually is interesting in a very specific way. It's not just another piece of physics that you need to know to track what cells are doing. It's actually giving us access to privileged access to a computational layer, a layer where decisions are being made about what happens, not just physics, but actually computation. Now, we took as our, in looking at this, we took as our inspiration the one system that is uncontroversially known to be able to scale up from individual cells to large scale goals, and that's the brain. So in the brain, you have a collection of individual cells, of neurons, and they can keep memories and they can pursue goals and so on. So how does the brain do this trick? Well, there's hardware, which is basically each individual cell has little proteins in the cell surface called ion channels. They pass ions such as potassium and sodium and so on in and out of the cell. And as a result, they acquire a voltage gradient. So they acquire a voltage potential across the membrane, and they can communicate that gradient across these controllable electrical synapses known as gap junctions. That's the hardware. The software, and you can see it here, this is imaging, a beautiful imaging by this group of a zebrafish brain, a living zebrafish brain thinking about whatever it is that zebrafish think about. And you can see all of the electrical activity. And it's a primary commitment of neuroscience that these electrical dynamics are holding the mental content of this little mind. That basically, if we were able to decode what the patterns were doing, we would know the animal's memories, the animal's preferences, and so on, right? So this is where all the software of cognition sits. Now, it turns out that the brain did not invent this kind of architecture from scratch. It was based on ancient principles that evolution discovered way back around the time of bacterial biofilms. And so every cell in your body does this. Every cell has ion channels. Most cells have gap junctions to their neighbors, and we can do a very parallel kind of effort to neural decoding in neuroscience, and we can do somatic decoding. We can say, I mean, here's a voltage sensitive fluorescent dye showing you the electrical activity of an early frog embryo. There are no neurons here, there's no brain yet, but already the cells are communicating electrically about who's going to be left, right, anterior, posterior, and so on. And you can imagine that if we understood how to decode, how to read and write this information, we would have some amazing control over growth and form. And so the first thing I want to do is I want to show you the tools that we use to kind of first to measure and characterize these things. So this is the voltage sensitive fluorescent dye, which allows us to see all of these things in real time. And then we do a lot of computational modeling and simulation. So once we know what the channels and pumps are in each cell, we can ask, where do these patterns come from? I'm going to show you a couple of examples of these patterns. So this is one called the electric face. So here's a frog embryo trying to put its face together. It's in time lapse. And what you see is this is one frame from this video. The depolarized regions are lighter here. And what you can see is that already before all the genes come on that are going to dictate where the eye is formed, where all the other organs, already this tissue has decided where everything goes. Here's where the animal's right eye is going to be. Here are the placodes. Here's the mouth. This is already, and I'm showing you this as opposed to other patterns because this is the easiest one to decode. I mean, it literally looks like a face. Right? We call this the electric face. It literally looks like that. We know that this pattern is absolutely functionally instructive, meaning it's required for normal development, because if you interfere with it, you create Picasso tadpoles. So you can move all this stuff around, move all the gene expression and all the anatomy by changing this electrical pattern. So this is a normal pre-pattern required. And of course, there are human channelopathies that have craniofacial defects that are due to disruptions in this process. Now, there are also pathological patterns such as this, where we introduce a human oncogene into this embryo. Eventually, it makes a tumor. Eventually, the tumor starts to metastasize. But before that happens, you can already see the bioelectrics traces of these cells disconnecting from their neighbors. In order to become individual amoebas that are going to metastasize and proliferate, they have to disconnect electrically from their neighbors that were binding them into a collective that was able to store large-scale goals, such as form a nice chunk of muscle or skin or something like that. Okay, so these are the patterns. Now we have to be able to, so we can read them. Now we have to be able to write them. So we basically just stole all the techniques from neuroscience. We do exactly the same thing. It's very interesting. The tools cannot tell the difference between neurons and non-neural cells. They work exactly the same, which tells you something interesting about our notions of what neurons are and how fundamental that distinction is. Exactly the same technologies work here. So I can take a piece of skin or liver or anything else, and I can control these electrical synapses by opening and closing them. So this is the topology of the network, who talks to whom. Or I can directly set their voltage by opening and closing these channels. So I can use drugs, I can mutate the channels, I can use light in terms of optogenetics. What we don't do is any electrical field application. So there are no electrodes, there are no electromagnetic fields or waves, nothing like that. What we do is use molecular physiology to target the very components that are used by this network to hold electrical properties. And when you do this, some very interesting thing happens. I'm going to show you some examples. One example is at the single cell level is this. I told you that when electrical connection between cells and their neighbors are lost, this is the first step towards tumorigenesis. And so here, what we do is we inject a fluorescently red labeled oncogene into this animal, and of course it makes a tumor, and there's the red, you can see that. But here it is again, blazingly strongly expressed, in fact, all over the place here, not just here, it's everywhere, and there's no tumor. And there's no tumor because we co-injected another channel, this happens to be a chloride channel, that forces those cells to be hyperpolarized and to keep electrically connected with their neighbors. So even though the oncogene is cranking, the cells are not going to go off and form tumors because they are electrically coupled to a network that overrides their individual cellular goals to proliferate, dedifferentiate, and metastasize, and keeps them harnessed towards a large scale goal. Now, what might those large scale goals be? Well, here's one example. I told you that in the electric face, we were able to get an idea of what the patterns are that kickstart the different types of organs. So what we did was we took an ion channel RNA and we injected it in a set of cells that are going to become gut. So here's a side view of a tadpole. Here's the eye, here's the mouth, here's the gut. And what we did was we imposed on a region of those cells the same voltage that normally would be associated with making an eye in the face. And sure enough, what they do is they make an eye. Okay, so what we've done is we've convinced these sets of cells that they should become an eye. Now, the so-called master eye gene, the transcription factor PAX6, doesn't do this. It only works up here in the anterior neuroectoderm, but the bioelectrics works anywhere. You can convince any cell in the body to start making an eye. These eyes have all the right layers, retina, optic nerve, all of that stuff. And so notice a couple of interesting things. One is that what we provide is basically a subroutine call. We don't provide enough information to tell those cells how to make an eye. In fact, we have no idea how to make an eye. Eyes are very complex and lots of cell types. We provide a fairly simple bioelectrical state whose interpretation is build an eye here. And then the cells do the rest. It's basically a subroutine call. We've identified a module that builds eyes, and we've identified how to trigger it where we want. And then it takes care of itself. So that's very good for regenerative medicine. You don't have to micromanage all the outcomes. There are also 2 layers of instruction here. This is a section through a lens that's sitting out in the flank of a tail somewhere that we've induced. The blue cells are the ones that we actually modified to induce this new ion channel. It's a potassium channel. Okay, these cells start to make an eye, but the whole system realizes that there's not enough of them to make a proper lens. And so what they do is recruit a bunch of their neighbors. All these brown cells here, they never got the channel. They're completely wild type native cells. And so there's 2 levels of instruction. We instruct these cells, you need to make an eye. They instruct their neighbors saying, come help us. The eye is supposed to be of this size, which again, we did not have to micromanage. So that's very good. We can make the same way by inducing electrical states. We can induce otocysts or inner ear balance organs. We can induce ectopic hearts, right? So extra hearts. We can induce extra forebrain. We can induce extra limbs, as I showed you here, and we can make fins. Now that's a little weird because tadpoles aren't supposed to have fins, and so we'll get to that momentarily. But you can see that by manipulating the large-scale bioelectrical patterns, you don't just change specific cells or cell differentiation into different cell types. This is a language at the level of organs, okay? And it goes beyond organs. It goes to whole body axes. So you can see here, this is a planarian. I take this worm, I chop off the head and the tail, and I leave this middle fragment. The middle fragment 100% of the time will regrow a head here, and it will regrow a tail there. How does it know where everything goes? Well, if you look, there's this interesting voltage gradient that says 1 head, 1 tail. Okay. Now, what we can do is we can manipulate the ion channels in these tissues to say, actually, I want 2 heads or I want no heads. Okay, so you can do that. We'll get into this shortly, so you can control now. Now, one thing here is that these heads are normal and they belong to this normal species. But you can go further than this, and you can ask a species to make heads that belong to a completely different species. So here's an animal with a triangular head. We chop off the head. We prevent the rest of the body from electrically communicating amongst the cells. So basically block the gap junctions for about 48 hours. Then we withdraw the blocker. The cells settle into a new electrical state. And sometimes they settle into their correct attractor and you get your normal head shape. But sometimes they make round heads like this S. mediterranean. Sometimes they make flat heads like this P. felina. These animals are about 100 to 150 million years of genetic distance, except there's no genetic difference here. There's nothing, we didn't do anything to the genome of these animals. This is purely temporarily, physiologically blocked them from communicating with each other. And they found states in the attractor landscape of that electrical circuit that belong to other species, okay, without any genetic change. And in fact, not only their head shapes change, but the shapes of their brain changes, the shape of the distribution and number of stem cells changes. You can go even further than this, and you can explore regions of the morphospace that biology doesn't use at all, and you can make planaria that aren't even flat. So they can be spiky like this, they can be cylindrical like that, they can be kind of a combination form. These cells are perfectly happy to build other things if the electrical pattern of what it is that they're building is altered. Now we can use this for biomedical applications. So here we are working on leg regeneration. Frogs, unlike salamanders, do not regenerate their legs. So you amputate, 45 days later, there's nothing. You can introduce a cocktail. The cocktail triggers a build a leg here kind of signal. Immediately you see there's pro-regenerative MSX1, it starts to come on. And within 45 days later, you've got some toes, you've got a toenail. the leg is touch sensitive and it's motile, so the animal can use it, and eventually it makes a very nice leg. The treatment here is only 24 hours. So what we do is we treat with the ion channel drugs for about 24 hours, and we don't touch it again. From a 24-hour treatment in adult frogs, these are froglets, but we've shown this in complete adults. In adult frogs, after 24-hour treatment, you get leg regeneration for a year and a half. Okay, so it really is a very early trigger where you convince the cells that instead of scarring, they're going to regrow whatever naturally is supposed to go there, which is a leg. And you don't get tumors, you don't get eyes, you don't get tails, you get proper frog legs. And they grow for about a year and a half without us having to do anything. Now, for biomedicine, we have to remind ourselves that yes, this is not some sort of frog specific or worm specific technology. We've done these things with human mesenchymal stem cells, with cardiomyocytes, all the kinds of things you would expect to do in vitro. What we're doing now is we're trying for limb regeneration in mammals. So the idea would be, can we use a bioreactor, a wearable bioreactor produced by our collaborators in the Kaplan lab. They make these bioreactors. Inside the bioreactor is a gel with ion channel drug payload. And again, only for the first day it's on, and then basically it protects the cells as they make decisions about what they're going to do. And I have to do a disclosure here because David and I have started a company called Multiscale Inc., which is dedicated to using these kind of approaches for regeneration of all kinds of organs, appendages and organs. Now, I should say that it is known at the single cell level how voltage changes talk to the genome. So the transduction machinery by which voltage changes in individual cells control gene expression, that's known. We know about at least half a dozen mechanisms by which this happens, some very common ones like calcium channels, some exotic ones like voltage sensitive phosphatases. So we know this. We know how the transduction works at a single cell level. And in fact, we know what's downstream. So we've studied using next-gen sequencing and microarray. We've shown that all your favorite BMP pathway, sonic hedgehog pathway, FGF, all of the WNT, all of these kinds of things are downstream of the bioelectric decisions that the circuit makes. Then you have to implement them by rearranging morphogens and so on. But all of this is, even though we know these molecular details at the single cell level, all of this has been highly unsatisfying because that isn't what we're trying to understand. We're not trying to understand individual cell decisions. We are trying to understand large scale, where large scale shape comes from and how to control it. And so what we really need to do is this, and so this is what we're working on, a kind of full stack approach, a multi-scale approach where we start with transcriptional circuits that result in specific channels being expressed. Then we model computationally all the bioelectric dynamics in the tissue that result from these channels operating. Then we can ask, how does this relate to large scale axial, sort of whole body patterning? And then from this, we can derive algorithmic kinds of descriptions of what's going on that are really suitable for manipulations. Once you have this kind of white box model where you can see what it's measuring, what decisions it's making, what are the control points, you can start to intervene. Intervening down here is really, really hard. And so this is a simulator called Betsy that our collaborator Alexis Pitak wrote that lets us take individual cells in a virtual tissue and model. Every cell has these networks, both chemical and bioelectrical, and then we can ask tissue level questions like, hey, when we cut it into pieces, how come every piece rescales the electrical gradient? How does that work? And so here's there's a model that could explain that. And so here's where the AI comes in. There's a couple things that we've done, and if people are interested, here's all the software. You can download it, and it's described in various papers, and you can play with it. Two things that are really important on the machine learning side. One is to help us infer models. So knowing the pieces that are in there, the various channels and transcription factors and everything else, what is a minimal model that explains the functionality that we see, that explains how it grows, how it regenerates, what shape it makes. So trying to discover models. And in fact, we've done this. So here in a few years ago, we showed how you can use evolutionary computation. So basically just simulate evolution over the space of possible models to try to match functional data of regeneration. So basically we had a system, we coded into it every known functional piece of data in the Planarian literature, so hundreds and hundreds of papers, not large data in the sense of like genomic sequencing, but actually functional data, like we cut it in a particular way, we treated it with particular drugs, and then look, it made, you know, a head and two tails or something. And so that kind of information goes into the database, and then there's an evolutionary computation across a simulator that tries to identify models whose behavior in the experiments matches what the database has in it from the publications. Okay, so it tries to infer action, and so this is an amazing. I'm still kind of amazed about the fact that this actually came up with a human understandable model of what's going on in planaria. You know, human scientists have been trying to do this for probably 120 years, and the system actually guessed a very nice model of how heads differ from tails. And that's the most creative part of the scientist's job, right? Is trying to come up with these models, and then here we have AI doing that. So I thought that was pretty cool. But we're going to have to have this because the amount of information, functional information, is flooding us at a rate at which no human scientist can keep all of these things in their head. So not only do we do this for inferring models, the next step is to infer interventions. So now that you have a decent model, you can ask this question. If I want the system to do something different, how would I intervene? Where, you know, which of these nodes and in what way do I need to intervene? And you can look for very rare kinds of interventions where most of them don't do what you want. And in this case, we were able to find some nice trigger points that let us control melanoma, basically the conversion of normal melanocytes into melanoma. So the AI is important both on the model inference and on the intervention inference. And to come back to the bioelectrics a little bit, because I want to get back to the medical aspects. One thing that you hear a lot is, I think it's important to understand the biology in a new way in order to enable all these things. And one thing I'd like to talk about is this metaphor of the code. So you will often hear that the DNA is the software of the body, and that actually the cells are the hardware that interpret the software. And I think that's okay for certain kinds of things, but I think it's profoundly incomplete and limiting. And I'll give you a different metaphor to consider. What I actually think is more valuable is the idea that what the genome actually does is it nails down the hardware of every cell. The genome is just a description of what proteins, including ion channels, are going to be present in the cell. But now, once you make a tissue out of these cells, even though they all have the same ion channels, you basically have made an excitable medium in which symmetry breaking and amplification can take place. And this is now a simulation, a model of that scenario where all the cells are proteomically identical. They all have the same channel. And yet, look at all this rich structure at the bioelectrical level. If you've seen Turing patterns, that's kind of the chemical version of that. Same thing happening in the brain. You can have all kinds of activity without having to wait for new gene expression to come on. And so basically, you can build circuits that have this interesting property, like a flip-flop, where they can store different electrical patterns in the same set of cells without changing out the cells or without changing the proteins in the cells, much like this flip-flop that can store a 0 or a 1 via the pattern of energy flow through the system. And just by taking stock of what the parts are, you can't tell what's in it. This is why bioelectricity work is so hard. You can't kill the cell and then fix it and then analyze it. All the information is gone. At that point, you have to study the living system and you have to do the physiomic profiling. It's all in the real-time electrical activity. It's not in the proteins. So if this is true, it makes an interesting prediction that we should be able to edit the software while keeping the hardware constant. And I've kind of shown you all that already in the examples of the eye and the heads and all that, but I want to drill down to one specific example. So here's a planarian, head and tail, has normal gene expression. So the anterior genes are in the head, not in the tail. And when I cut it like this, it makes a perfectly normal one-headed worm, as it's supposed to, and it's very reliable. Now here we have the same thing, one-headed animal, the anterior genes are in the front, so that's good. I cut that and I get a two-headed flatworm. Why is that? Because I just told you that it was reliable. How come the two-headed flatworm? And it's because what we've done in the meantime is edit the electrical pattern of this body to say not one head like here, but actually two. And when we do this, sure enough, it makes a two-headed worm. Now, here's the most important part of this slide. This electrical pattern is not a readout of this animal. This electrical pattern is the readout of this animal. In other words, this is not, the electrical pattern isn't what the anatomy is doing now. The electrical pattern is a stored memory, in this case, a latent memory, because it doesn't do anything until you injure the worm. It's a latent memory of what a correct planarian is supposed to look like, which gets consulted after injury. It doesn't do anything until then. And the bioelectric pattern does not have to match the current anatomy. Currently, it's a one-headed animal, and it can have a normal body can store at least two different ideas of what a correct planarian looks like. So I promised you at the beginning that we were going to find the set point for anatomical homeostasis. We're going to be able to read and write the memory of what the anatomy is supposed to be. This is it. And there are many more. I'm just showing you a nice simple example. We can now literally see how this tissue is encoding how many heads it's going to form if it gets injured. And that information can live in the same kind of body here. Now, one interesting question you could ask is, what happens if you take one of these two-headed animals and recut it in plain water? No more manipulations, leave the ion channels alone, I'm just going to recut it. So now the standard paradigm would tell you that, well, you remove the primary head, you remove this ectopic secondary head. All you've got left is a nice normal gut fragment. We didn't do any genomic editing or mutation, so surely all you're going to get is a normal worm now, right? In the absence of this crazy reprogrammed tissue, you're just going to have a normal worm. And that's, of course, what you would think, except that if you analyze the electric circuit, you see that it actually has two stable points. It has one stable point at the single head state, but it has another stable point at the double head state. And so when you do this, when you actually cut them, they don't go back to normal. They continue to regenerate as two-headed animals. So this system, again, remember, nothing has been changed about the genetic sequence here, and we can change it back to normal. We can take the two heads and convert them back to normal by blocking a particular ion pump for a couple of days. So this has all the properties of memory. It's long-term stable, it's rewritable, it's got conditional recall, and it has two possible outcomes. So think about what this means for biology. The question of how many heads this thing is going to have is not something that's nailed down by the DNA. It's not in the genome. What the genome gives you is a machine that reliably defaults to a pattern memory of one head, but you can rewrite that. And not only does the system keep it once you rewrite it, apparently forever, but it will obey the new pattern until you set it back to normal. So that's very important. That means you have hardware that has a default behavior, but you also have the software layer that you can interact with if you know how to read and write that information, that does not require you to do any genome editing. And so what we're trying to do right now is to really understand how the state space of these electrical circuits correspond to the different shapes, and especially to merge it with paradigms out of connectionist machine learning, where it's very understandable how electrical networks store patterns, how they store memories, and in particular, how they do pattern completion. When part of that information is gone, Hopfield nets and other things will restore it. And so there's a very nice kind of a merging of dynamical systems and energy minimization and learning and information. And so I think biology has tons to learn from the kinds of things that machine learning has been working on. Okay, so in the last couple of minutes, I want to tell you about where I think this is all going. And first, I just want to show you something about the rational design of so-called electroceuticals. This here is the pattern of a frog brain, the electrical pre-pattern of a frog brain. And you can see the important thing to see here is that there's a particular pattern here that tells the brain exactly how wide it should be. So it sets the size and shape of the brain out of this whole, this whole entire tissue. And it has a very particular bioelectrical pattern. And the thing is that this bioelectrical pattern is critical, because if that bioelectrical pattern goes wrong, and it can be affected by nicotine, by alcohol, by various teratogens, by mutations, all kinds of things. If that pattern goes wrong, what you get is birth defects of the brain. So here are the eyes, here's the forebrain, here's the midbrain and the hindbrain. You can have a hindbrain that kind of looks okay, although not great. And then the midbrain is just completely screwed up. The forebrain is just gone. The eyes end up connecting directly to the midbrain. It is just terrible. So we asked a simple question: if we know what the correct pattern is, and we know that the pattern has become incorrect and thus led to this defective craniofacial structure, what would we have to do to change that? So we made a model, and this is again work with our collaborator Alexis Pytak. She and Vaibhav Pai in my group made this model of the electrical circuit of the early brain and what goes wrong with it. And then they asked the simple question. They interrogated the model to say, okay, if that's what's wrong, what channel would we need to open or close to get back to the correct state? And notice that what you can't do, so here's this bell curve pattern is what you want. You can't just raise everybody up to be at this level, because that gives you brain defects. And you can't drop everybody down to this level, because that also gives you brain defects. You have to try to recover this particular pattern. Now, that's very complicated, but the model was amazing in that it told us that there's one particular channel called HCN2 that would actually do this. It would leave these cells down low, it would raise these cells up high, and it would predict that it would recover this. Amazingly enough, and I'm always shocked when these models actually give you something that works to fix these very complex defects. It actually works. Whether you introduce new HCN2 channels or you use drugs to open existing HCN2 channels, here's a normal brain, here's a defective brain. In this case, it's even worse than a teratogen. These animals have been a mutant form of a notch protein has been introduced. Notch is a very important neurogenesis gene, and without that, the forebrain is gone. The midbrain and hindbrain are a bubble. These animals have no behavior; they just lay there doing nothing. It's a really strong defect. Despite the notch mutation, look, the structure of the brain is back, and in fact, their IQs come back. So their learning rates, you can test their learning rates and so on in behavioral assays. The learning rates are indistinguishable from normal. So you can rescue even not only from teratogens, but actually from really nasty genetic defects if you manage the bioelectrical pattern correctly. And the way, so this is important, this was not a screen. We didn't try 100 different or 1,000 different drugs. The model told us very specifically which channel we would have to open to get the right bioelectrical pattern. So we've moved, when I started this work in 99 or so, 1999, we were at the point of doing screens, just asking what are bioelectrical states for? What do they do? And now we're to the point where in certain cases, we can make a model that specifically suggests therapeutics. It tells you which channel drug to use to fix a very complex defect. And so the vision I see going forward for biomedicine looks something like this. There's a simulator that takes as input expression data, which already exist from RNA-seq studies of what channels are present in what cells. Those are your control knobs. Those are the things you get to tweak. And of course, what bioelectric pattern is desired for your tissue. Now, this is largely missing. This needs to be acquired. These physiological data sets need to be acquired in most cases. But once you have those two pieces of information, you can ask the model, what channel and pump would I need to open and close to get the right pattern? And then you can simply look for existing channels. Something like 20% of all human drugs used drugs are ion channel drugs. And then you can simply look for either develop new ones or look for them. And so this is, again, we've already, you can go play with this a little bit. We've already started on a basic framework here where it's called the electroceutical design environment, where you would sit down, you would pick the cells and tissues that you want. You would say, my pattern is wrong. What do I do? And this thing would actually then tell you which drugs you actually need to use. Okay, so that's kind of the vision, and all of this part is the machine learning part, because interrogating those models is not easy. You can't do it just in your head. You have to do the simulation, and you have to be able to work the simulation backwards through a search of some sort to know what the intervention should be. So just to close here, here are my conclusions. That there's a very important physiological layer of software that sits between the genotype and the anatomy, and it's a really tractable target for new biomedicine. Evolution discovered very early on that electrical signaling is a great way to process information, to integrate decisions across distance. So the fact that we have organs and tissues that scale correctly and that connect up to each other is a consequence of that fact. We now have the ability to read and write some of these pattern memories, and to reprogram large scale shape, heads, eyes, appendages, not by micromanaging, not by controlling stem cell properties, but by telling the collective what it is that it's supposed to be building, right? Taking advantage of the fact that it is a homeostatic system. And that new machine learning tools are coming online as a kind of robot scientist platform to help human scientists infer bioelectric and biochemical circuits from functional data, and then to infer interventions, right? So how do we manipulate those circuits to give us the outcome that we want? And I think two things will come from all this: not only fantastic regenerative medicine approaches that will do way more than I think is actually doable with classical, let's say CRISPR and things like that, but will also actually feed backwards and help us design new AI platforms that are not based on brain architecture, but on much more ancient principles of computation and scaling of cognition in early cells. So that's it. I want to thank the students and the postdocs who did all this work. Thank our funders. Thank the model systems who actually do all the heavy lifting for all of this research. And again, a disclosure: there's a company called Multiscale Inc. that David and I are part of. Yeah, and I thank you for your time, and I'll take questions.
Hello. Thank you, Dr. Levin, for that talk. I know a lot of our attendees signed up for our symposium just to hear it, and I'm certain that it lived up to their expectations. It was truly revolutionary, and I had no idea there was such a complex bioelectrical layer that controlled our physiological outcomes. Now we will have a 10-minute question period. So if anyone in person has questions, I will ask them to please line up here at the podium. And any online attendees, as before, may put their questions in the chat, and one of our execs will relay them to Dr. Levin here. Speak into the microphone, please.
Hello, doctor.
How you doing?
Sorry, I was just curious that when you mentioned that about inducing bioelectric signals with ions to make changes, is more or like is additional energy required from elsewhere to fuel the changes in development, like say addition nutrients and minerals? Is there like an order of complexity to it?
Yeah, interesting question. I don't think so. Now, nobody's measured this, so it's possible that there are subtle differences, but I don't think there are major differences, because what you're doing is you're not necessarily introducing new proteins. You're not doing anything other than opening and closing the existing channels that are there. And so I think, well, what it does require is a baseline level of resting potential, which is kept up usually by the sodium potassium ATPase, and so that has to be kept going. But I think not much energy is actually used to open and close these channels. So as far as I know, I don't think there's an energy limitation here. If anybody's talking, I can't hear anyone. I think you guys are muted. Oh, there we go.
Just press that, because it's just on at this time. No, you.
Okay, can I?
Speak.
Okay. Professor Levin, thank you so much for the talk. It was really fascinating.
Thank you.
So I don't have much of a biomedical background, so all of this looks like magic to me, much of it. But I do have a computer science background, and I'm trying to figure out how AI could help in this endeavor. And so from what I understand, the models that you're speaking of could involve like looking at a bunch of data that you collected from all of these different animals, and then finding out some patterns which can then extrapolate to new kinds of data, make new predictions, that sort of thing. So is that the kind of models that you're implying, or do you have something else in mind? Could you maybe say a few words?
Yeah, so there's been a couple of different types of uses of AI here, and then we can talk about what's still missing. The thing that we did in Planaria was to basically build a database, basically build an expert system that knows every published result in Planaria research. So every experiment of the form, I did this and then I saw that, I did this and then I saw that, right? So these, experimental setup result, right? So those those kind of pairs, we have hundreds, I think like 800 maybe in the database. So that is a knowledge database that's everything we know about Planaria. And then what the machine learning has to do is using a simulator, so a virtual planarian, it has to try to come up with a model of the regulatory circuit within each cell, such that when you pull them together into a tissue and you start doing virtual experiments on them, give the same answers as what's in the database, right? Most models, of course, don't match that. And so this was an evolutionary algorithm that eventually found a model that acts exactly like not only did it act like the experiments in the database, of course, we held back a bunch of experiments that it never got to see, and then it correctly predicted those experiments, and then it actually gave us some that had never been done before that we did, and we saw the right outcome. So one thing you want to do with machine learning is to infer models of what's going on, not just predict numbers and that kind of thing, but actually infer human understandable descriptions of what the algorithm is that it's operating inside the cells and also across the whole tissue. So that's one kind of thing. Another thing sounds more like what I think you were describing, which is that if we had a large number of bioelectric measurements across different organs, different ages, different types of animals, then you could ask the system to try to infer patterns in that data and try to help us guess the bioelectric code. That would be super cool, and I think we should do that. The problem is that typically machine learning requires large numbers of data. These experiments are very difficult to do. They're very time consuming. We do not have anything like the volume of data that's required for typical approaches in this field. So I think until the technology catches up, that kind of thing is going to be pretty hard. But nevertheless, what we have also done is apply machine learning to models of the code that we do have. For example, the brain. This is how we discovered the intervention that fixes the brain, and ask the machine learning to pick interventions given an existing model. So still lots of technology to be developed, but I think already we can see how it's going to be used.
Okay, now we will take a question from the online chat. It says, how does the evolutionary process work for the cells to know the correct bioelectric patterning to produce the ultimate anatomy?
Yeah, great question. I mean, where does the pre-pattern come from in the first place? So we've tracked this down. The best example we have of this is in the early of left-right patterning in the frog embryo, because we can track it all the way down to the single cell, the fertilized egg, and so we have all that information from the beginning. The best way to explain that is I'll tell you the physics side of it, and then I'll tell you what I think is the kind of computational side of it. The physics side has to do with symmetry breaking and self-organization. Basically, much like Turing patterns, where you can have very specific kinds of patterns appear in a well-mixed medium, if you have ion channels expressed across a sheet of cells, and they're all identical, so no patterning has taken place yet, there's no pattern, everybody's the same, pretty soon you're going to have very characteristic patterns. In the Turing patterns would be spots and stripes, and bioelectrics it's slightly different, but pretty soon you're going to have patterns spontaneously arising, and what determines the shape of those patterns are fundamentally the property, two things: the properties of the ion channels and the laws of physics. That's what determines the shape of the patterns that spontaneously arise in tissue. And so what evolution does is optimally tweak the properties of the electrical of the ion channels and of specific signals that get kicked off at different time points by various cells that optimally shape those patterns such that they end up producing a useful organism. Okay, so that's kind of the physics end of it. You can see that, and it's kind of using conventional concepts. There's another way to think about this, which is my students often will, when they first get introduced to this, they'll say, but where is the pattern recorded? Like, I see the electric face, where was that pattern recorded? And I have to remind them that patterns don't have to be recorded. So for example, I can take a couple of transistors and I can make an AND gate, and it's a piece of physics, but it also does logic, and it has a very nice truth table associated with that. Or maybe I build a calculator and it can do sines and cosines. And then you can ask the question, where is that truth table encoded, right? How does it know how to do that? And again, we have to come back to the fact that all of these things take advantage of the laws of physics and computation. If you produce a specific kind of physical machine, it's going to have behaviors. This is the software part. It's going to have behaviors that are not necessarily directly encoded anywhere by anybody. It's interesting, various interesting laws of physics working through this machine. And so that's the other way to think about this. Much like with computers, you end up with all kinds of calculation, interesting calculations and computations that don't have to be explicitly sort of provided in the genome or anywhere else. That's not where they are. They're like kind of like the properties of mathematics. So yeah, so those are the two ways of thinking about it.
Thank you, Dr. Levin. We still have many people lined up for questions, but we want to be respectful of your time. If you have 5 more minutes, we could take maybe 2 more questions and then we'll finish.
Sure, let's do 2 more questions. And anybody that I don't get to here, feel free to drop me an email, and later, I'll get back to you offline.
Okay, thank you very much.
Another planaria related question here. So you mentioned that when you cut the original head off, that another, it could potentially grow the head of another species. Would that mean that the head part would be genetically different from the body part?
No, the genetics are completely intact. What it tells you is that the genetics don't determine the shape of the head. And we already know that, right? We shouldn't be, anybody who uses computers should not be really shocked that the same piece of hardware can do multiple different things. So there's a default, right? So any kind of, like, think about a programmable calculator. You turn it on, and by default, very reliably, the first thing you see is 0. That circuit on the little LCD screen, so that circuit has an electrical state that it reliably starts off in. But it's a programmable calculator, which means that you can now give it stimuli or inputs that alter the way that the pieces talk to each other internally. And then you can cause it to remember various things and to do things that it wasn't doing when you first turned it on. Same thing here. The genetics produce cells that by default reliably end up making triangular heads in that particular species. But the cool thing about that hardware is that's not the only thing it knows how to do. That's just what it typically does. You can, by judicious interventions, you can basically push it into different regions of the state space of the electrical circuit that genome knows how to make. So it's all of this stuff is forcing us to rethink what is the relationship between the genome and the anatomy. This idea that the genome somehow codes for the anatomy is that, that's just not the right way to think about it. The genome codes for a machine that by default knows how to make that anatomy. And by the way, if you want to see a really kind of a egregious example of this kind of thing, I can go to my website, and under the presentations, there's a bunch of talks about xenobots. And we show that skin cells with a perfectly normal frog genome can build a whole other type of creature. It's kind of a synthetic proto-organism, has different behaviors and different shapes, and does all kinds of things. It's the exact same genome. So we got to just keep that in mind, that the genome does not directly set the shape at all.
Thank you.
All right, I had a question about the methods you're using to change the electrical patterns.
Mm-hmm.
Why are you not using electrical fields? And are you solely using drugs or genetic manipulations of changing how much ion channels are expressed?
Right. We use drugs, we use ion channels modifications, and we use optogenetics, so light. So why are we not using electrical fields? For the following reason. There's nothing fundamentally wrong with electrical fields. In fact, back in the day, starting in, I think the earliest experiment was like 1903, when people didn't have these other technologies, that's what they used. They used electrical fields. The problem is that electrodes and applied electrical fields are really good at two things. They're good at spiking excitable cells. So if you want to trigger action potentials, that neuroscientists do all the time, they're great at that. So you can, put in a like kilohertz kind of a thing and spike neurons. That's fine. And they're good for providing vectors for cell migration. So if I put two electrodes across a piece of tissue here, the cells will feel the electric field. Most cells love to crawl in electrical fields. They're just going to start crawling. Okay, though that you can do. But notice what we have here. What's important here are the stable, steady state, long-term distributions in resting potential. That means I need cells over. So in the electric face, I need cells over here that are -30, and they need to sit that way for hours. I need cells over here that are -80, and then some more -30, and then some -20. So it's this complicated pattern of resting potentials that you can't set that up with electrodes. If I put electrodes on either side of a cell, what happens is this side of the cell gets depolarized, that side of the cell gets hyperpolarized. The net voltage of the cell does not change at all. There's no way that I know of to use electrodes to set up a stable, steady, long-term complex image or a pattern of electrical potential. It's just not doable for now. Maybe someday somebody will invent the way to do it, but for now it can't be done that way.
Okay, thank you, Dr. Levin. So lastly, for anyone whose questions are still remaining, could I ask you to put your email in the chat so that people can see it?
Yes, absolutely. Yeah, great questions. Thank you so much. Thank you for the opportunity to share these ideas. That is my email address. Please feel free to drop me an email and we'll chat.
Thank you very much.
Bye-bye, everybody. Thank you. Thank you so much.